Okay, so as you see, let's see. Well, I guess we were very good in the sense that 57% of the people got the right answer. So we're going to be learning a lot about the drugs that have been approved today, but also about ones in clinical development that are really exciting. Can I have my first slide, please? As Ann pointed out, the clinic has always been near and dear to our heart, and that's why back in 2004, Kathy Giusti founded the Multiple Myeloma Research Consortium, and as you've heard today, it started from 4 sites, it's expanded to 22 leading centers, and over the course of the last 11 years, the consortium has done 70 trials with 35 different agents and really work to speed those drugs into the clinic. And could I have I don't have my slide pointer. And what we pride ourselves in the consortium of doing is to really have a balanced clinical pipeline, to make sure that we're offering different types of drugs that target specific molecular alterations, drugs that are of novel classes and from novel signaling pathways. And again, you're going to hear a lot about that in the next section. And in this section, we're really going to focus on immune therapies. And immune therapies have been, as Ann just pointed out, very exciting in the cancer field. It's been like a renaissance of immune therapy over the last decade, where there have been exciting approvals in other cancers with patients of high unmet medical need in melanoma and lung cancer. But as we heard, there have been approvals most recently in myeloma of 2 different antibodies. And as you'll hear today, there are many different types of immune therapies that are being studied clinically in myeloma. So at this point, what I want to do first is to call up our panelists that it's a pleasure for me to introduce. I'd like to call up David Avigan. He's a professor of medicine at Harvard Medical School, and he's also active staff in the Hematology Oncology Division, bone marrow transplant at Beth Israel Deaconess Medical Center. I'd next like to call up Dr. Raphael Kleins. He is a scientist and group medical director at Bristol Myers Squibb. And finally, I'd like to call up Dr. Alex Lisokin. He is an assistant attending for the myeloma service at Memorial Sloan Kettering Cancer Center in New York. So thank you so much for joining us here today. And you have real experts on this panel in immune therapy, so really welcome. And you're going to hear a very exciting panel discussion. So let me just start off with that. We do have an arsenal of immune therapeutic approaches. Again, 2 antibodies, Darzalex and Implicity, approved last year. We've heard it was a great year for myeloma patients. 4 drug approvals, 2 new antibodies. And we'll be describing these antibodies to everyone today. We also have another class of antibodies that will be described, the immune checkpoint antibodies. And Dr. Kleins will go into it in more detail, but tumors really know how to mask themselves, protect themselves from our immune cells. They've really evolved to evade and avoid the immune system. And it's these immune checkpoints that actually help unmask the tumor cell, take off the brakes of the immune cells, so the immune cells can then recognize and destroy myeloma cells. So those are the antibody therapies that we're looking at, but there are also, and those will be reviewed by Dr. Lisokin, there are also different cellular therapies like vaccines. We know about vaccines. We get vaccinated, and it helps prevent certain diseases. Well, these vaccines that we're talking about in oncology is not necessarily vaccines that help prevent the cancer, but they actually help to treat the cancer. How can you do something to trigger a patient's immune system to be able to respond to the myeloma that's growing inside of them? And also, another type of cell therapy is, how do you take a patient's immune cells, like T cells, engineer them in different ways reinfuse them back into patients, so now these engineered T cells can then recognize the myeloma cells. These are all different cellular approaches that Dr. Avigan will be talking about. The MMRF really considers immune therapy of essence to what we do as part of our portfolio. And therefore, what we have created is an immune portfolio. And as you can see from this immune portfolio, it represents our end-to-end solution of the 3 pillars in terms of building a bigger data set. So as you've heard, and I think in the last panel it was mentioned, that not only are we going to use COMPASS samples to be genomically profiled, we're also going to use COMPASS samples and other samples we've collected in other initiatives to be profiled to look at the different types of immune cells that are present in a patient and whether they're functioning, to better understand what a patient's response is to immune therapy. This we're building, so we're going to build the immune component of COMPASS in a big way to then give this information back to researchers so that they can start to ask complex questions, come up with hypotheses. In terms of the learning network, we also have a portfolio. We've invested in last year in translational research in immune therapy. Translational meaning that portion of research that goes from the bench to the bedside. It's in between. It's preclinical studies. And we gave 2 MMRF grant awards, one doing work in CAR T-cell therapy, and that was given to an investigator at City of Hope, and another award that was granted to UCSF to study a drug that's conjugated or bound to an antibody that could then go and recognize myeloma and destroy the myeloma. So that's preclinical work. In addition, Dr. Avigan's going to talk to you about his vaccine trial. That's a very exciting trial. I believe it just opened today, yesterday, very recently. And we actually supported some of the research years ago in vaccine studies in his laboratory. And we are supporting the research for this study that he's doing in his lab that correlates to the clinical trial. So those correlative studies that will help us understand why some of these patients will respond to the vaccine and why others might not. And then as we move in from the learning network and we learn about all these findings, what are we doing in the clinic? Well, we have a very exciting collaboration with a large foundation called Cancer Research Institute that focuses on immune therapy, and we're proud that we have this collaboration because it's enabling a clinical trial that Dr. Lisokin will talk about, which is a combination of 2 different immune checkpoint antibodies. And finally, I'll just get to the point, the MMRC has done a number of different trials in immune therapy. There are several of the investigators here today with us from the MMRC, and so we have, as part of our portfolio that you just saw, a focus on immune therapy. Antibodies, we have a very exciting trial testing immune checkpoint inhibitors right now, and we will be adding more and more to our portfolio. So with that, I am going to turn this over to Raphael. Raphael, you can either sit at your seat or
I can sit, that's fine, but I need this. Well, thanks, Joan, and thanks to the MMRF for inviting us here. I've been asked to introduce 2 of my friends and colleagues, David Avigan and Alex Lisokin. We share some common roots at Memorial Sloan Kettering doing tumor immunology over the years. I'm now at Bristol Myers Squibb for the last 2 years, having drank the proverbial Kool-Aid to get involved in bringing nivolumab or PD-1 therapies to more and more patients with cancer. And the future is bright for this activity in myeloma. David is pursuing vaccine approaches that should marry very well with a checkpoint inhibitor that would release a brake on his T cells that he induces with the vaccine. Alex is looking at other methods to drive the immunity or the immune response in the patient, for instance, by killing more of the tumor cells with radiation and enabling these new antigen spreading phenomenon to induce more T cells that, once again, maybe at the very end needs this checkpoint inhibitor to enable the T cells to do their final job. So it's a real collaborative kind of experience to induce in patients, these immune response that take different pieces of the immune cycle to get things going. We're catching that at the very end of patients whose tumors have evaded the immune system and have developed multiple reasons why they've evaded the immune system. We've been lucky enough to find in the last few years this major checkpoint inhibitor that by itself can help 15 to 40% of patients, but not all patients, and we need these additional steps to bring the therapy to induce responses in more patients. So this picture sums up a little bit of what we are thinking about in myeloma. There's a cell at the top there that's got 2 nuclei. It's got no reason to be surviving. And in fact, Jonathan spoke a little earlier that one of the checkpoints in the tumor biology is p53, and maybe this cell has a broken p53 system that prevents this cell from doing what a normal cell will do when it has this kind of major aberration. That's to commit suicide. And so this tumor is allowed to grow despite the cytogenetic mishaps that have accumulated along the way. And it's sitting in a sea of immunity. The bone marrow is an immune organ. And around those cells around the tumor cells, the sort of fried egg cells, are also cells that have very different morphologies. They look very different. And these are immune cells that are interacting with the myeloma cells. There's one very dark cell that might be a lymphocyte. That's not doing anything. It's actually, you can see it's not activated. It's a dark, resting nucleus. So we need to reawaken these lymphocytes that are probably recognizing, some minority of them are recognizing tumor but not finishing their job. And so we should be thinking about how to induce immunity by driving cell death of these tumor cells to drive more and more T cells to get activated, and then PD-1 might be the finishing touch that's needed to release the brake on the final killing. So this is a new way of thinking about cancer therapy. For 40 years, we've been doing something quite different because the hypothesis, which is true, that cancer is a loss of cell growth, leads to a different sort of idea of how to treat it. We would need to give therapies that kill lots of cells. It kills them more quickly than they divide. So the more therapy, the better. And that is the basis, or it went to its logical conclusion in high-dose therapies for solid tumors, for cytotoxic therapies and radiation. And then we learned what are the drivers of this aberrant cell growth, the Achilles heels, and now we can use molecular changes to try to reimpose cellular brakes on those tumor cells to stop them from dividing. And that's led to very important advances to inhibit tumor cell growth, targeting various pathways that you've learned about a little bit today. And then there are epigenetic therapies that also can change what's called the cell cycle, the ability of the cell to grow. So these are good things, but there's whole other evolving landscape here, not just an evolution of evolving cell growth, but also evolution to evade the immune system. And therapies that we've been trying for years to introduce into cancer, to re-educate the immune system so that the tumor can be seen, make it more immunogenic. And then the checkpoint inhibitors are really a way to remove brakes on the final killing of the cells. And so we do this in 2 different ways. One is to provide patients with the molecules that can do the killing, antibodies, and both elotuzumab and daratumumab are in this category. Engineered T cells that are taken, grown out of the body into high numbers that all have a specific receptor that recognize only myeloma cells, reinfused back into patients. And then things that we're calling engagers that are kind of like antibodies. They recognize a tumor antigen on the surface, but they also bridge on a second arm to the T cell. So it brings the T cells to the one of the early steps that were taken for immunotherapy of cancer were monoclonal antibodies that brought together not T cells and the target, but immune cells, NK cells and macrophages that bind this antibody that binds the target. So the antibody targets the cell and the immune effector cells kill it. And in the 90s and 2000s, 5 tumor types had antibodies approved for cancer. And of course, it's a phrase that's often used, the magic bullet for cancer appeared very promising for many cancers. And when this was written in 2001, it took 15 years for myeloma to get what it should have gotten all along, is tumor targeting with antibodies with daratumumab and elotuzumab. In November of 2015, both drugs were approved. And these are very effective medicines that can induce remissions in most patients. And now we will begin to use them in combination with other therapies to maximize their effectiveness together with the existing drugs with myeloma. The other approach is not to use antibodies, but rather the other arm, T cells. And the major advance for many solid tumors, now 6 tumor types, we have approval for checkpoint blockade, melanoma in 2011, and 5 others this past year and a half. And I'm just going to show you, let's see if I can figure it. Can you click on the black box? Is that possible? It's a little movie. So what I'm showing you there, the little cell is the T cell. He's going to kill the bigger cell. He's injecting those granules, and then the tumor, in this case a normal cell, is turning into a dead cell. It looks more like it's kind of a raisin as it's done. Maybe one more time. So this was a virally infected cell. The T cell sees the viral antigen, injects those little black dots, and that kills the cell and shrinks the tumor. I didn't know it had sound. That makes it a lot better. So now in cancer, that tumor cell blocks the T cell from killing. The engagement happens, but the tumor is in stealth mode, and it upregulates, it expresses molecules that prevent the T cell from killing it. It's a don't kill me signal. And the antibody, this checkpoint blockade, relieves the don't kill me signal so that the killing can take place. And it needs to be brought into more and more cancers. And over the past year and a half, it's been introduced in clinical trials in myeloma. The middle is those adoptively transferred T cells, those engineered CAR T cells that everyone's been hearing about. This one happens to recognize New York ESO1, that's the antigen. And the bottom panels are from left to right, I think it's left to right. Left to right, you're seeing accumulation in red dots. Those are all T cells in the patient over a month after treatment. And that coincides on the top with the loss of the red dots, which were the tumor cells. As you get more and more killers, you get fewer cancer cells. And these are engineered to express one specific TCR, so it is relatively safe in that New York ESO1 is not really expressed on normal cells. And so the checkpoint universe is broad right now. In myeloma, 15 active checkpoint studies in myeloma. Below it is just the example for melanoma, different tumor, but was the first approval for combination immunotherapy was in melanoma. The 3 curves are on the bottom are the progression-free survival, how long the patients live without their tumor progressing. For ipilimumab in green and for nivolumab in blue, 2 different checkpoint receptors. And then the combination on the top. And it's obviously an example where 2 is better than 1. It tells us that these combinations, these different approaches, when combined, should work better than when they're given alone. But it tells us we have a lot of work to do, to think about which ones should be better, to do work to substantiate that rationale, and then to do the clinical trials. So we've had a doubling. There's a lot of interest in immunotherapy and cancer overall, and in myeloma, it's true as well. So a doubling in the number of active clinical trials in the past year across these 4 areas. And there are 600 combination trials going across all cancers currently ongoing. So on the top are 9 different immunotherapy agents that are currently being explored in early clinical trials at BMS. And so that gives us 9 different opportunities to combine with nivolumab or ipilimumab. And of course, there are combinations you can give think about between the two of these novel 9. So it's a lot of opportunity, and we need good science to back this up. And so the MMRF really does provide a venue for the community to bridge this gap, to do the work, to try to substantiate which of these combinations might make sense, using existing drugs in the armamentarium for myeloma on the left, combining with these various active and passive immunotherapies so that we can treat both the tumor and the patient. And the COMPASS itself is a unique database to try to drive rational choices, because already we have a description genetically of all these tumors. All these greater than 1,000 banks, this huge biobank of more than 1,000 tumors genetically described. And now, associated with that, in the bank, in the freezer right now, are the viable immune cells of each of these patients that are waiting to be studied. So we can ask, if patients who have RAS mutations or MAF mutations, do they have more or less immune responses? Do they have more macrophages or less, more Tregs or less? All these specific questions give us some potential answers for how we would treat these patients. And COMPASS had the foresight, the MMRF had the foresight to put them in two separate buckets so that we could first define the genetics of the tumor, and then we can define how that genetics influence the host response. And it'll categorize patients not only for genetic status, but associated immune status, obviously giving hints to which genetic lesions we should hit to change the immune response. And importantly, it's people. And in this room, there are people who are very happy to come here, work with the MMRF, because they do such a great job, and the results are, as described earlier today, phenomenal in terms of what this foundation has done for this disease. So it incentivizes all of us to work with them so that we can share in this progress, both industry partners and friends and colleagues from academia. And so with that, I'll be happy later to answer questions with Alex and David and hand the
So thank you, Raphael, for the great introduction. And now I'm going to turn it over to Alex to talk about antibody treatments, and then after that to David Avigan to talk about cell-based therapies. What I just want to ask both David and Alex is to maybe interject. We've heard from Raphael how COMPASS samples could be used to learn more about the immune system. Well, as you're speaking about trials and some of the exciting new trials that you're working on, what you're including in those trials for us to understand why patients are responding to different immune treatments.
So thanks very much for the introduction. I think Raphael actually went through a very good intro of what we're trying to do to drive an immune response. Oftentimes when we try to explain this as, scientists or what have you, we can make it sound like a textbook and be very confusing. So I like to speak here in analogies that when we're driving an immune response, we have a T cell that interacts with the tumor cell. That is like a lock and key kind of thing. It turns the car on, so to speak, but the car just sits there until it gets further instruction. You got to press the gas, you got to press the brake. So we now know what a lot of these gas pedal molecules are. We know what a lot of the brake molecules are. Those are the checkpoints that you've heard about. And I think particularly in myeloma, we need to understand what drives, what turns the wheel. And I think, COMPASS will help us with that. But we also have a variety of drugs, IMiD drugs, the directed monoclonal antibodies, vaccines, and CAR T cells that you've heard about and you'll hear more about, that are potential drivers that help us get the car going where it needs to go. So when we talk about immune checkpoints, this is, if I go to an academic meeting, this is a slide I see in pretty much every single talk, so I felt like I had to include it here too. Basically, this is a whole family of molecules. Again, these are the gas pedals and the brake pedals. On the right of the slide, you have all the brake pedals, and these are all targetable. They can be blocked with an antibody drug, and a lot of these are being targeted clinically. And on the left, you have the gas pedal molecules, the things that drive the cell this is the T cell, what we think is the important immune cell that drive that cell to continue to proliferate, continue to survive, continue to do what its intended function is. And so you can design antibodies actually that target those molecules and trigger them to be more active. We call them agonists. And so you can turn up the activating, you can block the inhibiting, and by doing that, you can further stimulate a T cell to do what it's designed to do, which is to eliminate its target. The trick is that you need that steering wheel. You need the T cell to be there. You need the T cell to know what to target. And this is what I think David and I have discussed. We'll be talking about what kind of things can we do to do that. So one thing that can be done is to essentially label cells. And monoclonal antibodies like anti-CD38 antibodies like daratumumab or like elotuzumab, they can identify the myeloma cell, but in a way that antibody can serve as a label that the immune cells can see, and that can trigger an interaction. And in fact, some of these antibodies, particularly anti-CD38 antibodies, can also modify the environment which the myeloma cells can live in, that the myeloma cells live in, to enhance the ability of a T cell to do what it wants to do, which is to kill the myeloma cell, what we want it to do. And so these antibodies, of course, increase responses. They're in some ways are thought of as risk agnostic in terms of the genetic heterogeneity that was raised earlier. And they have a very favorable side effect profile. And they offer a really unique potential for immunotherapy combinations. And I think the MMRF and MMRC are involved in some of these combinations of these types of studies. And so I think we, as Raphael has said, we've been focused a lot on killing the plasma cell, and I think that's actually incredibly important. It's led to huge advances in how well, and how our patients do. This is something that the MMRF has been involved in for quite some time and has really moved the needle in. And as we think about molecular targets and things like that, that's also very important. But we can also think about ways to help the patient attack the plasma cell. And you'll notice that some of the drugs that are on the left side of the slide are also on the right side of the slide. Because a lot of the drugs that we currently use can be viewed from an immunologic lens. And so this means that we have the tools to drive the car, to turn the steering wheel towards the plasma cell. And we just have to learn how to make those combinations. So IMiD drugs, monoclonal antibodies like daratumumab or elotuzumab, combined with immune checkpoints is a very rational combination. And these are things, there's already a study out there that the MMRC is involved in, and there are other studies that are in development, looking at next generation checkpoints as well. And so the idea is that you activate that happy looking T cell to go after the plasma cell. Another way is to build on stem cell transplant. So stem cell transplant is something that is routinely used in myeloma, as many in the room obviously know. But this is also an immunotherapeutic manipulation. We are, through our treatment, we're able to, we routinely have all of the logistics set up to remove cells from the patient to give cells back. Well, we can manipulate those cells when we remove them for a CAR T cell study. Well, we can also condition those cells with a checkpoint, take them out, do a transplant, and put them back in where there's a lot of space for them to expand and to really immunologically target myeloma. And so this is a study that has now been a couple of years in development, but actually is about to open in next week, actually, that the MMRF has collaborated with and supported together with the Ludwig Institute to combine two checkpoints together with transplant to try to sort of take the autologous stem cell transplant and make it almost like an allogeneic like transplant, to turn it into a very immune, essentially an immune therapy, to try to get that durability that we can see with an allo transplant into auto transplant. And I think with that I will stop.
So, Alex, what do you study? Are you studying different responses in the patients associated with the immune system?
Yeah, no, for sure. And of course, as we design these kind of studies and think about these studies, a critical aspect of them is the correlative analysis of what we think we are targeting the myeloma cell, but we really need to show that we are targeting the myeloma cell. And a lot of the studies built into this are using very similar sort of genetic pipelines that have been already discussed at the previous session, where we want to analyze genetically the myeloma cell, analyze genetically the microenvironment, try to understand what the immune cells can see on a myeloma cell, and to really find out, do they see it? And what is the outcome of that vision? Will it lead to a more durable response? And I think that those are the critically important studies, which I think we wouldn't really be possible without support similar to support that we've got from MMRF to do.
Well, thanks, Alex. And I just want to comment that one of the trials that Alex was referring to was a trial we're doing in the consortium. It's with a drug, a checkpoint antibody from Genentech. It started out just with their checkpoint antibody and in combination with Revdex. And about a year into the trial, Genentech decided that they wanted to expand that study, and in collaboration with Janssen, decided to add additional arms. So now they're looking at their checkpoint antibody in combination with DARA. So that trial expanded, and there's a lot of excitement. It's great to see two companies with different drugs being able to work together with us as well as our institutions to get this trial up and going. But it'll afford definitely new investigational options to test in myeloma patients. So with that, I'm going to turn it over to David, who's going to talk to you about cellular immunotherapy, how you can manipulate a patient's immune system. So,
thanks, Jan. So
he's done wonders with that.
It's a real just privilege and such an exciting thing to be here. This is really a very special time in myeloma research, as I think we've all been hearing all morning. And I think we're all really confronted with this reality that therapies that we didn't even know existed are now available to our patients. There's a real impact on patients' lives, both in terms of their responses, their quality of life, the sort of overall prognosis. But we still run up against, in many patients, a difficult situation where resistance over time develops. And the question is, what can we do with the immune system that would be unique or special to try and eradicate disease in a more effective way? And importantly, see what we can do to try and prevent recurrence, which is such a obviously big worry for all of us. Why the immune system? What is sort of unique or special about it? Well, we know over millennia, our immune system has evolved to recognize what is foreign and does not belong, and differentiate it from self. So there is this sort of opportunity, if you could harness this power, to selectively target malignant cells, if you can show them to the immune system as being foreign, and go after them in a way that is different, frankly, than chemotherapeutic or targeted therapies. Because there is the ability to selectively target things that are not dependent on just specific pathways. And so you use the immune system's ability to target a large population of cells and to broadly target those cells. And so we know that one of the sort of great challenges in cancer therapy is the heterogeneity of the tumor, the flexibility, or we call plasticity of the tumor to adapt, and critical subpopulations that exist within that tumor, such as the malignant stem cell population. Those populations in some ways can all be targeted immunologically. And so one can look towards an approach that would recognize that heterogeneity and essentially go after it. And the second critical part that cellular therapy at least has the potential for is the idea of memory. We know that when we give a drug, the drug is present, has its effect, and then it's gone. And that's, of course, part of the therapeutic window of the drug. The immune system has the ability to remember. And so when we vaccinate against mumps or measles, we're not just sort of trying to prevent an issue that month or that year, but we're creating an army of T cells that have the ability, or other what we call effector cells, to circulate and provide ongoing protection. And so when we're thinking about cellular therapy, what we're really trying to do is, number one, educate the patient's immune system to see the tumor as foreign and to provide ongoing protection in that realm. And so that is a lofty, difficult, but incredibly important goal. Now, if you're going to wade into this in myeloma, one of the things we have to be aware of, and this is true in cancer in general, is that the immune system undergoes certain changes that allow the tumor to emerge. And there's been a lot of careful research on this over many years with many investigators showing that there is a gradual erosion in T cell immunity that goes along with the progression of disease from early steps, monoclonal gammopathies, to smoldering disease, to then more clinically active disease. That is paralleled by a sort of gradual dysregulation and loss of T cell immunity. And so our quest is, can we essentially repair that, restore it, and would that essentially create long-term remissions? So we're going to talk about two strategies, one that has been near and dear to our hearts, which is the cancer vaccine strategy. And frankly, I have to say, would never have gotten off the ground without the support of MMRF and other foundations, because this was a bit of a cockamamie idea that when we were initially starting, we had sort of a genesis of a new sort of approach, but did not have really the proof of principle yet. So the basic idea here of a vaccine is that, as was mentioned, tumor cells learn to evade the immune system by showing themselves as essentially belonging. They hide. And they do this with upregulation of the negative checkpoints, by not presenting antigens properly, by essentially blending in as if they were a normal skin cell. On the other hand, the immune system has certain teachers whose job it is to essentially flag cells as foreign. And so dendritic cells, which are these sort of very most powerful and champion teachers, have the ability to present antigens or to present proteins in a way that show the immune system to go after them. And so the first idea of a vaccine was, let's harness these cells to teach the immune system to see cancer as foreign. The second idea is, well, are there targets that we could accentuate in a tumor that would make it clear that it's foreign, that are different from normal cells? And we know that there are. There are ones that are called shared antigens, and there are also ones that are called neoantigens, antigens that are derived from mutations that drove the tumor to be created to begin with. And so this is a little bit of a bridge between what we talked about earlier with precision medicine and the idea of genomics and targeted therapy and immune therapy. The idea that by understanding the mutational landscape, we can identify targets that we could accentuate to the immune system to go after. And so we have both a necessary teacher, the dendritic cell, and the ability to then highlight targets within the tumor. So what you see here is our vaccine platform, which on some level is kind of simple and crude. We don't know what the most important antigens are in an individual patient, and it may not be the same in every patient. So what we decided to do is to take the patient's own tumor and create a personalized vaccine out of it by taking that tumor and all of the different heterogeneous populations that make up that tumor and create a fused cell or hybridoma that links it physically with dendritic cells we generate. So now you have this cell that expresses all of the proteins of that particular tumor, now presented in the context of this immune stimulating cell that teaches immune cells to see it as foreign. And this was an idea that was first developed in animals and in preclinical work, where it was a fantastic way to cure mice of cancer. But it's not exactly necessarily the same story as when you get into patients. And so we went into a series of trials, first phase 1, and I'm not going to labor just because of interest of time, but I want to just talk about briefly a phase 2 study that we did, which is sort of keeping in what was just mentioned, the idea of using a transplant as a platform to reduce disease, but then the immune system as a way of going after what's left. And so what we did is we took patients who were going through an autologous transplant. We know that those patients tend to get into good responses, usually very good partial responses. And then we vaccinated them right after their transplant. Now, you might think that's kind of a dumb idea, because if the immune system is beat up after a transplant, how is it going to respond? But what we found, and this is where details are very important, what we found is actually the immune system is quite altered in the post-transplant period. And during that period of recovery, some of the strategies that the tumor cell uses to hide are broken. And we say tolerance is transiently broken because you have essentially the emergence of myeloma-specific T cells that all of a sudden appear in the blood that were not there prior. They're there in small numbers and they disappear over time. It may be that some of our maintenance therapies, like using lenalidomide in that setting, actually takes advantage of that population. But what we showed is that when you vaccinate people in that post-transplant period, you can cause a dramatic expansion of those myeloma-specific T cells, and they hang around and persist. And what we saw in this initial study is that our complete response rate nearly doubled from day 100, we think after the chemotherapy effect was done, to a year out. So this was exciting and provocative, but if we want to be grown-ups in this field and like test things in a very effective way and not in a boutique way, we needed to explore, can we actually test this in a randomized trial in the same way we would for a drug, and can we do this with the larger community and make this available to a larger number of patients? And so we did something that was a little bit crazy, maybe in keeping with the theme of being disruptive and bold, which is that we presented this to the cooperative groups and said, we don't want to just do this in our single center and sort of treat, a group of 30 or 40 patients. We want to see if we can do this over several hundred patients in a randomized trial. We will invite people from all over the country to come to Boston, and we will teach them how to do this. So we generated a trial that involved people from all over the country working together. We invited everybody to our lab, showed all of our SOPs, and in an open sort of format, just basically taught everybody how to do this. And so we have 15 sites from around the country who are all participating, most of the major cancer centers around the country. It was kind of humbling because you invite people into your kitchen and show them exactly how you do stuff. And of course, people start saying, really? Because we started doing it that way and that was kind of a bad idea, and then we started doing it this way. And in this sort of interaction, we actually really learned a lot. And we had to kind of be humble because people were kind of making all kinds of suggestions that you can imagine. The most amazing things, we got 15 centers to all agree to do something together. But it happened. And so just to point out the critical point that the MMRF played in all this, which is that we wanted to not just study this, which is now going to be randomizing a few hundred patients after transplant to get lenalidomide alone or lenalidomide plus our vaccine, but we wanted to understand what's going on. So with the partnership for the MMRF, we have designed a complex set of scientific correlates that will be looking at each step of this. And so it's not enough to see, okay, what's the effect? That's critical. But can we predict who is more or less likely to respond? What is the nature of what we're doing well and not well? What other pieces can we add to this? And so we have science that's being done at our lab about T-cell immunity, at MD Anderson for NK immunity, humoral immunity that's being done at Emory. Roswell Park is doing our MRD work. We have a group in Nikhil and the Farber's lab that's doing some MRD work as well. So we brought together people in a way of doing this in an integrated way with the MMRF support. So I think that, and the study started about 2 weeks ago, and I've been spending all morning on my email trying to actually deal with the first series of patients. So it's really exciting. People are really into it. And I would just say that this platform, which we hope makes a huge impact on myeloma, is not just about myeloma. This exact platform that we're looking at, we are exploring in leukemia. We just got some really exciting data that we're putting together where 70% of our patients who were treated in first remission have not relapsed now 5 years out. And that's also now going to go into a large multi-center randomized trial. The last thing I want to sort of just mention is the other side of the equation. So we talked about the idea of educating T cells inside the patient. And what about the idea of taking T cells from the patient and programming them without any need for education? And this is sort of one of the strategies that has been pursued as CAR T cells that Carl June and Michelle Sadelain and others have kind of pursued, and also engineered T cells, which are Aaron Rappaport and others have different strategies to basically take the T cells out of the body and let them see a tumor as foreign. Like everything that we're doing, it's all about the details. It's all about getting greater biologic understanding. This is an incredibly effective strategy in acute lymphocytic leukemia, shockingly effective in patients who are otherwise refractory to other treatment. And for those of you who might have seen that Empire of Maladies movie, there's an incredibly poignant story of a young girl with a resistant disease who is now doing okay 5 years later and has met Obama and Lady Gaga and a bunch of other folks. How does this apply to myeloma? And I think it is very important to say that the immune environment is different in each cancer. As people have been commenting, we need different strategies in the same way we don't use the same chemotherapy for every type of disease. So I think we need to understand that if we're going to use engineered T cells in myeloma, we need a good target, and people are exploring that. People initially looked at CD19 because it was available to us from the work that was going on in leukemia and lymphoma. That may or may not be the perfect target, but there are others that people are looking at. When you turn T cells on, they can really kill, but they can cause toxicity. So one has to look at how to regulate that. So there's a lot of exciting work, but the basic principle of turning a T cell on and killing cancer that is not killable with standard chemotherapies is something we've already shown in principle across a couple of different diseases. And now how to integrate these things together is going to be a real challenge. So let me stop there and I guess give time to make sure that everybody has time to ask questions.
Yes. So first of all, I want to encourage our participants to be submitting questions through their iPad. And I just want to ask the panelists here if they could just speak up a little more, because it's being live streamed. So I just want to, let me kick off the questions and say, so Alex and David, you've talked about antibodies, immune checkpoints, you've talked about cell-based. We know that myeloma, we need combinations. So what are your thoughts in terms of combining cell, immune checkpoints, DARA, implicity, whatever? I'll let you comment.
Okay, first, sure.
David, or?
I jump in first. So I think that, I think as Raphael said, the idea of a checkpoint inhibitor, for instance, is that you have a T cell present with the capacity to recognize and kill a tumor, but it's being restrained. It's being tolerized, it's being turned off, and so we're trying to undo that restraint. Of course, that only will be effective as a single approach if the T cell's there. And we know in the most sort of florid way, a disease like Hodgkin's disease is notable for having lots of T cells sitting at the tumor site that don't do anything. I think we all feel in a disease like myeloma, we have to first expand that repertoire of T cells. Whether that's through an education process, which is what a vaccine is, to basically take T cells that are not really expanded, expand them, and then further activate them, for instance, with a checkpoint, with an IMiD, with a hypomethylating agent, different ways that can affect that. Or whether to actually engineer the T cell outside the body, we talked about some of those approaches, and then activate them. I think CARs, for instance, are also subject to being tolerized and downregulated. And so one has to think about clever strategies to both create your response, activate it, and sustain it. So I think it's a relatively, at least, straightforward idea, of course it has to be tested, that these things will fit together in an effective way from a strategic point of view. And we are, in our group, looking at that. We've actually just completed a study with our vaccine and one type of PD-1 modulating drug, but who knows what the best one is and what the best sequence are and whether other kind of combinations, as was talked about, are going to be important. The only way to know is to actually do the studies, but the rationale of putting these together, I think, is strong.
And maybe Alex or Raphael, maybe in terms of combinations, also consider combining not only immune, but immune approaches with other types of approaches, targeted approaches, epigenetic approaches. Maybe you could comment on that.
Yeah, for sure. No, I mean, I think...
And if you could just speak up a little, Alex.
Yeah, so I would echo everything that David said, is that, you need to have the T cells present. The checkpoint is, in a way, and the agonists are the ways that drive those T cells to do their function. And in addition to vaccines, which I think are, very, very important, and sometimes in the euphoria surrounding PD-1 blockade and CAR T cells forgotten, and they should not be. And I think it's like a testament to the tremendous efforts that David described, that this is going forward in myeloma in this very exciting randomized study. But in addition to vaccines, in something called in situ vaccination, where you're not really vaccinating by delivering something that you've made through an injection, but where you are in effect killing the tumor in an immunogenic way, in a way that facilitates having that tumor cell, the myeloma cell, present itself to the immune response in a way that isn't hiding it, but in a way that is facilitating greater T cell education and T cell response. So in situ vaccination strategies, things like radiation, things that combine epigenetic modulations that sort of can turn up the amount of things that a cancer cell would express to the immune system. Things like chemotherapy, melphalan, which is routinely used, that can kill myeloma cells, things, and IMiDs and antibodies, other strategies. These are all things that can be used to help educate the immune response, and combinations with checkpoints are a very relevant thing to pursue in that context. I would also say that, these sort of combinations are going to take time to develop, and they need to be done in a stepwise fashion to really make sure that you can do things safely. So, for example, you've heard about a vaccine study that is transplant-based, where you deliver the vaccine afterward. You've also heard about a study that uses that same sort of transplant to try to test whether or not we can deliver checkpoints, immune checkpoints, after transplant. When you have that initial, breakage of tolerance, can you further drive that with immune checkpoints? Ultimately, it makes complete sense that if that's a viable, a strategy that can be safely applied, that these things could be combined to further enhance each other and to synergize. And CAR T cells could also work into that. So, but these things need to be done in a stepwise fashion, and I think they're on all of our minds. And each one of us, can't do everything, so we do our different parts, and then we get together and we talk, and then we figure out the next step forward.
And in light of that, what about using immune therapies like the antibodies in maintenance?
So, I don't know if
Raphael, do you want to?
The antibody therapy should presumably work across all stages of myeloma, since this mechanism, all it requires is expression of the appropriate target and the presence of the immune cells. So I guess it's plausible to think that in a minimal residual disease setting, the job is not as, the hurdle is not as huge. So it certainly should be tested. The maintenance is a place where we want to extend people's, complete responses for years, hopefully forever, and obviously give non-toxic therapy. For some of these antibodies, it looks like the combinations are really still required, including an IMiD plus an antibody. And that is actually not a surprise given the in vitro data, the experimental data that preceded it. I think combination of an IMiD and an antibody makes sense in a maintenance strategy. And, at some point, we'll also probably see checkpoint inhibitors and IMiDs making their impact there as well.
So I just want to say there's only a few minutes remaining, and I encourage all of you to find me, or if one of our panelists at the end, to answer some of the questions that we couldn't answer today. But what I'd like for everyone, all three of you, to do in 30 seconds or less is to come to some type of summary or conclusion of the direction of immune therapy for myeloma. Alex, do you want to start?
Sure. I mean, I think we have a lot, I think it's a very exciting time in immunotherapy for all diseases, and specifically in myeloma. We just have gotten, 2 FDA-approved antibodies that help label the cells. We already have a lot of experience with IMiD drugs that can facilitate immune responses. We know how to use radiation in this disease. We know how to use transplant, and I think it's just a matter of taking all of this knowledge and putting it together to really facilitate the long-term sort of durable responses that we hope for with meaningful immunotherapy.
Raphael?
Yeah, I mean, I think, immunotherapy of cancer has been, 30, 40-year ambition. And until 5 years ago, there really wasn't the data that would tell us we need to work redouble because we're getting somewhere. The PD-1 story has changed that. In more than 5 cancers where we have, exceptional response rates. In myeloma, the drug by itself is not effective. It taught us that we need combinations. So it teaches us that we have more work to do in this disease to drive immunity. But we know some of the factors. These 40 years of learnings have taught us a lot. We need T cells in the tumor. We need interferon signaling. We need mutations. So we will be able to sort of come up with a laundry list of things we have to drive to make PD-1 work.
And finally, David?
Yeah, I mean, just to echo, this is an incredibly exciting time. I think there's a lot for us to do and accomplish. The intimate link between scientific understanding, patient involvement, foundation involvement, sort of the tie between industry and academics, I think have all brought us to a very important point here. I think as someone who takes care of patients, we also have learned a fair amount of humility that things happen in a way that you have to be careful and thoughtful and stepwise, that you don't go beyond your sort of situation where you're not doing things in a careful, thoughtful, planned way, so you understand where you are. And I think that that's really been the theme of the day here, which is how do we take these rapid advances, move the ball forward, and at the same time understand where we are and do right by our patients. So I think that there's a real opportunity for that now.
Well, that's great. So I just want to thank the 3 of you for joining this panel today on a very exciting and promising field.
All right, thank you so much. Really a very complex field, and thankfully Joan teaches graduate courses in immunology, so she takes that and teaches all of us at the MMRF as well, because it really is complicated. And as you can see, most of these strategies are not yet ready for prime time. I was even a little bit surprised that over 40% of the folks in the audience weren't sure which antibodies were approved for myeloma, and those are the proven strategies. Forget about all of these other ones that are in very early clinical development, but does say to me that I think there are some things that we at the MMRF can do in terms of some of our educational programming to make this a lot easier to understand. So the broad takeaway, again, think of immune therapy as also being down the road precise. It's up to us in the work we're doing to figure out what are the right treatment strategies for every single individual and to do those correlative studies. And again, the correlative studies that we've supported since the MMRC started, trying to understand who responds to which therapies and why, so that we can make smarter decisions in the future.